The undead genes. How can death tell a story? Dead bodies can't talk, or can they? Not in the traditional sense, and not in the undead sense either. Their bodies, whether the cause of death was natural or otherwise, hold information in their very DNA that can help an investigation into the circumstances leading to their death. DNA is the building block of life, and in order to create life, the instructions, the genes, need to be read and translated into a form understandable by molecular machines. This is a process called gene expression, scientifically defined as the process by which information from a gene is used in the synthesis of a functional gene product. During life, gene expression varies as a result of many conditions, including stress and sleep. Curiously, however, even after you die, your cells undergo a very unusual activity. Some genes actually increase their rate of gene expression, as seen in a study of 1,000 genes in mice and zebrafish undertaken by microbiologist Peter Noble at the University of Washington. It is not fully understood why genes do this, especially as the genes involved include early fetal development genes and genes related to cancer. Scientists from the Centre for Genomic Regulation in Barcelona have recognised the potential that this phenomenon holds in a number of avenues of research. Today, we will be focusing on the forensic application. Calculating time of death is crucial in a criminal investigation and could lead to identifying an assailant, or in civil investigations such as insurance fraud or property inheritance. Traditionally, estimating time of death is complex and relies on circumstance and pathological signs such as body temperature, rigor mortis, and lividity. More in-depth techniques are required for a more accurate estimation, and this is where the twilight of death holds potential to change the way that death is investigated. Roderick Guido observed the thanatotranscriptome in a paper published by Nature Communications entitled The Effects of Death and Post-Mortem Cold Ischemia on Human Tissue Transcriptomes. In this paper, Guido and his team looked at thousands of samples from various tissues to determine how death changes human gene expression and whether this information can be used forensically. RNA sequencing data of more than 7,000 samples from 540 donors were used from a reference database called GTEX. This data included a key number, the PMI. The PMI, post-mortem interval, is the time in minutes from the moment of death until preservation by the GTEX procedures. In comparing individual tissues to gene expression, it was found that there was a strong association between time since death and expression. In some tissues, this is more evident than others. See Figure 2. For example, in the muscle versus the brain tissue. This tissue specificity of response to death is incredibly useful for further investigation. Guido and his colleagues categorized and observed the main temporal patterns of change in functional activities after death. See Figure 3. Of the 5, the way that carbohydrate metabolism is affected is particularly significant, as it suggests that hypoxia is likely playing a major role in the initial pre to post-mortem transition, and this can be used for future analysis of cause of death. Using the data from GTEX and their initial findings on tissue-specific patterns in the transcriptome, a machine learning model was created in order to predict the PMIs. Their model learned the gene expression patterns of 399 people and tested it against other samples in order to hone the predictions. It must be noted, however, that the predictions are slightly more complex than the simple monotonic changes with PMI, but this model has accounted for covariates, but more can be done in the future to make this model more accurate. Their model was overall proved to be effective in predicting the PMI of 129 individuals. See Figure 4. In the beginning, they often found that the models tended to overestimate PMI, but concluded that if all the tissues consistently predict very similar PMIs, the prediction of the PMI for the individual is more reliable than if the tissue PMI predictions are very variable across tissues. They showed that RNA sequencing performed on a few key tissues can predict an accurate estimate of time since death, and a protocol was written to aid future forensic scientists and pathologists in harnessing these findings. In conclusion, Geiger and his teammates have kick-started the investigation into other, more specific methods of determination of time of death and opened the doors to less conventional ways of investigating crime. Not only can time of death be predicted with reasonable accuracy, but cause of death may also hold similar tissue-specific expression patterns, which would benefit from further analysis on larger, more diverse sample sizes. Alongside forensics, this study also has a lot of potential in the realm of transplantology, providing an alternative perspective to the transfer of the organ from donor to patient through upregulation of regulatory and immunological genes.